So we're back, and it's my pleasure to introduce Eric Skaar, professor of pathology, microbiology, and immunology at Vanderbilt University in US. He obtained his PhD in immunology and microbial pathogenesis from Northwestern University, and his MPH in epidemiology and biostatistics from Northwestern University. Sorry about the mispronunciations. So Professor Skaar holds several titles, serving as a director of Institute for Infection, Immunology, and Inflammation, and as a director division of molecular pathogenesis. And he also holds an Ernest V. Goodpaster Chair in Pathology and is the Vice Chair of Research Department of Pathology, Microbiology, and Immunology at Vanderbilt University. So Eric's current research interests are factors and processes involved in the battle for mettle between bacterial pathogens and their hosts. So I think that's a fascinating topic of interest. And today's presentation is the intersection of nutrition and infection at the host pathogen interface. So please welcome Professor Skaar. And now you can start your screen share. Thank you.
Thank you for that introduction. And I will make sure I can, let's see. Great. All right, thank you very much for the introduction, Siga, and thank you for Siga and Eva for putting together this meeting. I'm sorry we all can't be together. I've never been to Iceland. I was very much looking forward to the opportunity to go to Iceland. I'm hoping I'll find another reason to get there sometime soon. So,
hope so too.
Yeah. So as Siga mentioned, my lab is interested in this intersection between nutrition and infection. And the nutrient that we primarily study are nutrient metals, which is the reason for being here at this conference today. And the reason for this is because, of course, we all know that all cells require metals in order to carry out basic physiologic functions. Bacterial pathogens are no exception to this rule. They have to acquire nutrient metals in order to colonize vertebrates and cause disease. And this is something that the human body and all vertebrates have taken advantage of throughout evolution by becoming one of the most metal-starved places on Earth from the standpoint of availability. So, of course, we have plenty of metal in our bodies, but we have these immune processes in place to sequester metal and keep them away from invading pathogens. So all organisms that can make us sick have to have strategies to acquire nutrient metals inside of our bodies, and those typically take the form of high-affinity metal acquisition systems. We're becoming increasingly appreciating this concept that in addition to competing with the immune system, microbial pathogens have to compete with members of the healthy microbiome for these nutrients. And then finally, there's old and as well as emerging data suggesting that diet can affect these processes. And so I put this slide up as an introduction to my talk, but it's also an introduction to my research program. So my lab is interested in how bacterial pathogens acquire nutrient metals during infection. We're interested in the immune strategies to sequester those metals. We're interested in this three-way interaction between the immune system, pathogens, and the microbiome, and then we study how diet affects those processes. We primarily do this in three separate pathogens. We study Acinetobacter baumannii as our pneumonia model of infection, and we study Clostridium difficile or Clostridioides difficile as our gut model of infection. And we study Staphylococcus aureus as our systemic model of infection. And Staph aureus is the organism that we will talk about primarily today. So Staphylococcus aureus is the #1 cause of skin and soft tissue infections. It's the #1 cause of infectious heart disease. It's a primary cause of food poisoning, although of course we're all paying a lot of attention to emerging viruses right now. It's important to remember that some of these antimicrobial resistant bacterial pathogens still cause a tremendous amount of morbidity and mortality. And we're very interested in staph from a nutrient acquisition standpoint because it's a little bit unique in the bacterial pathogenesis community because it can colonize essentially every tissue in a vertebrate host. And that poses some very interesting nutrient acquisition challenges because the organism has to have different strategies depending on the organ that it is in. So in the first part of this talk, I want to tell you how we identify host factors that restrict nutrient metal from invading pathogens. And this work was performed by the people on the bottom of the screen, Brian, Thomas, and Victor. were postdocs in my laboratory, and Yao Fang was a staff scientist, and Jessica was a PhD student in chemistry. And I have to introduce you to the technology that we use to identify these proteins, and it's called imaging mass spectrometry. This was developed by Richard Caprioli at Vanderbilt. And the way that the technology works is that you have a tissue section, and you coat it in this case, a multi-matrix, and then a laser rasters over the tissue section. And you end up with mass spec data that can then be converted across 2 dimensions into an image on the tissue section. So I like to think of it sort of like immunofluorescence or immunohistochemistry, but you don't need any a priori knowledge about the molecules you're imaging. Instead, you just have the mass spectrometer tell you what molecules are there. And this can be a very powerful discovery-based technology. So here is an example of a single experiment imaging a rat brain, and each thumbnail is a different molecule that has a different distribution pattern throughout the tissue. And then you can zoom in on these thumbnails, and you'll see that different molecules have different molecular distributions. And then based on these distributions, you can, of course, generate hypotheses. And so we became interested in this technology when we first started collaborating with the Caprioli lab. And at the time, they were primarily using this in cancer biology. So we started a program with Richard's lab applying imaging mass spectrometry to infected tissue or to study infection biology. And the animal model that we predominantly use for this is a murine model of systemic Staph aureus infection. So we infect the mice retroorbitally or intravenously. And 96 hours after infection, we harvest the tissues, sacrifice the animal and harvest the tissues, and we see the formation of these abscesses, which are the hallmark lesions of a Staph aureus infection. And we're looking at infected kidneys in this image. And these macroscopic lesions make really excellent tissue for which to apply imaging mass spectrometry. And so here's an example of the kind of data that we can obtain. This is images of S100A8 and S100A9, and you can hopefully tell that they localize perfectly to the staphylococcal abscesses. There's a few words about these proteins. S100A8 and S100A9 heterodimerize to form the functional form of the protein called calprotectin. I know Professor Nolan is presenting after me, and I'm expecting that she'll show us some very beautiful work on this protein as well. Calprotectin is fascinating to me, particularly for this fact. It is approximately 50% of the protein composition or constituent of the neutrophil cytoplasm. So 1/2 of all the protein in the cytoplasm of a neutrophil is calprotectin. And if that surprises you, it's probably because most people, when they think about neutrophils, think about the granules. And that's where most of the antimicrobial proteins are. Well, those granules are bathed in this calprotectin protein. So I think it's safe to say that calprotectin is the most abundant protein in neutrophils, which arguably makes it the most abundant immune protein in the body. In a paper in Lancet in 1990, calprotectin was shown to be highly inhibitory to a variety of fungal and bacterial pathogens in vitro. And the model for that antimicrobial activity was through nutrient metal sequestration. But there were only 2 real pieces of data at the time to support that model. The first was the structure of the apo form of the protein had 2 predicted high affinity metal binding sites. And the second was that you could reverse the antimicrobial activity of the protein with excess metal. So bacteria plus calprotectin, the bacteria don't grow. Bacteria plus calprotectin plus metal. In this case, in the case of these original experiments, it was zinc, and the bacteria would grow well. But there's a caveat to these studies, and that is that they were entirely performed on 2 organisms, and they were Borrelia burgdorferi, which is sort of famous in the metal world. I'm sure many of us know because it has evolved to not require iron and can substitute in manganese and zinc for some of its iron-requiring enzymes, and therefore has a higher zinc requirement than some bacteria. And the second is Candida albicans, which is, of course, a eukaryote, and eukaryotes have a higher zinc requirement than bacteria. So we wondered at this point, knowing that calprotectin accumulates in the staphylococcal abscess, what might this protein do to an organism like Staphylococcus aureus that's not known to have a particularly high zinc requirement? So in collaboration with Walter Chazen's lab here at Vanderbilt, we purified functional heterodimeric calprotectin and tested its antimicrobial activity in vitro. This is a bacterial growth curve looking at optical density on the y-axis over time on the x-axis with increasing concentrations of calprotectin. And hopefully you can notice that at 75 micrograms per mil, calprotectin completely eliminates the ability of Staph aureus to replicate in these experiments. And, I think an interesting question at this point is whether or not this is a physiologically relevant concentration of calprotectin. And this is one of my favorite tidbits about this story is it's probably not a physiologically relevant concentration because calprotectin has been measured in abscess tissue in excess of 1 milligram per mil. So there's probably way more calprotectin in a tissue abscess than what we have in this experiment, suggesting that the protein is probably inhibitory to growth in vivo. We next became interested in whether the metal binding properties of the protein were important for its antimicrobial activity. So we mutated those two predicted metal binding sites, which we call site 1 and site 2. And we showed that this protein doesn't bind metal using NMR, and that it folds properly using NMR and doesn't bind metal using isothermal titration calorimetry. And then we tested its antimicrobial activity, and we saw a 90-fold reduction in the antimicrobial activity of the site 1, site 2 mutant, demonstrating that not only is the protein antimicrobial, but it must bind metal in order to be antimicrobial. So at this point, we became interested in whether or not this protein was involved in protection against infection in vivo. So we went back to our murine model of infection that I introduced you to, and we infected a calprotectin deficient animal to ask the question whether or not that animal was more or less susceptible to Staph aureus. And one thing I didn't point out when I showed you those early images is that the kidneys are predominantly the tissue that becomes abscessed in the murine model. For reasons that are not clear, most other tissue in the mouse does not become abscessed in a systemic model of Staph aureus infection, which was very different than what we saw when we harvested organs from the calprotectin deficient animal. And here you'll see the livers of these animals are highly abscessed compared to a wild type animal, demonstrating significant pathogenesis caused by Staph aureus. And if we homogenize those tissue and enumerate bacterial burdens, we see about a log more bacteria are in the livers of the calprotectin deficient animals as compared to wild type. So these data show that calprotectin is antimicrobial in vitro and also required to prevent colonization and abscess formation in the liver in this murine model. And at this point, we wanted to try to determine whether or not this ability to protect against infection was due to the metal binding properties of the protein. And so to do that, we went back to some imaging technologies, and I was very fortunate many years ago to meet Jörg Feldman at the University of Aberdeen, and he introduced me to the technology of laser ablation inductively coupled plasma mass spectrometry, or LAIC-PMS, which is a lot like the imaging mass spectrometry that I've introduced you to so far, except instead of using a MALDI to image proteins, you can use an IC-PMS to image elements. And so we began applying LAIC-PMS to infected Staph aureus infected tissues to image how infection affects elemental distribution. And here we're looking at calcium, manganese, and zinc in these experiments. And I just want to draw your attention to three things. The first is that calcium is an outstanding marker of the tissue abscess. We have hundreds of images now, and everywhere we see an abscess, we see large amounts of calcium. We think that's probably because of the robust infiltration of calcium-rich immune cells to the site of infection. The second is we see this really beautiful distribution of manganese in the uninfected or mock-infected kidney that's disrupted in the infected animal, and we are not sure what the biological processes are that are responsible for that massive rearrangement of metal in those tissues or metalloproteins, but we're very interested in trying to figure that out. But the question we were most interested in, of course, was whether or not manganese and zinc, at this time, those were the elements that we were interested in, are localizing with the abscess, and whether or not calprotectin has anything to do with that localization pattern. So we'll zoom in on another abscess here to show these data a little bit more closely. First, we'll show iron and copper, which in these experiments didn't have any particularly unique distribution patterns, but you can clearly see that iron and copper are very sparse inside the abscess, which is in contrast to what you see with magnesium and calcium. Magnesium and calcium accumulate in the abscess, and if you look closely, hopefully you can tell that there appears to be a pattern to that distribution within the abscess that's mirrored between magnesium and calcium. And this was interesting to us because it suggests that instead of being this big ball of amorphous pus as an abscess is historically characterized, there may be some molecular architecture within the abscess, and I'm going to show you some more data as the talk goes on that will support this idea. But at this point, manganese and zinc were the elements that we were most interested in, and we saw that manganese and zinc were occluded from the abscess. In data I'm not going to show you, we found that calprotectin was required to at least remove manganese from the abscess in these animals, and then we also see this sort of hard shell of metal around the outside of the abscess, and we are not sure what is responsible for the formation of that, but again, very interested in trying to identify the molecular features that associate with this metal shell around the outside of the abscess. So at this point, I'll just summarize and say that once we showed that calprotectin deficient animals are more susceptible to Staph aureus infection, a number of groups have shown that in other infection models that the calprotectin deficient animals are more susceptible to a variety of fungal, bacterial, and parasitic infections. But this is not the entire story, as some pathogens seem to benefit from calprotectin. So Salmonella typhimurium can acquire nutrient metal more efficiently than members of the microbiome. So when inflammation occurs in the gut and metal is sequestered, Salmonella can thrive, whereas its competitors do not do well. Helicobacter pylori senses alterations in the metal environment, leading to changes in the outer membrane that make it invisible to the innate immune system. And Streptococcus pneumoniae, it's not totally clear how the organism benefits, but it does benefit. So I'm going to switch gears right now, and instead of talking about calprotectin, I think we're very fortunate to have Professor Nolan present after us, and she's done really beautiful structural and biophysical work on that protein, and I expect that we're going to see some of that. So I'm going to stop talking about our contribution to the calprotectin story, and instead acknowledge that Zooms can be kind of hard to sit at home and watch, especially depending on the time zone you're in. So I wanted to focus the rest of my talk on something that's a little bit easier to look at, and that is going to be imaging. So here what I'll do is tell you about how we've been applying imaging mass spectrometry to infectious diseases. The team involved in this on the top are from my lab. You've seen Yaofeng and Jessica, Jim and Ahmed were postdocs in the lab, and Michelle was a graduate student. And the team on the bottom is in John Gore's lab, who is the director of the Vanderbilt University Institute for Imaging Sciences. And I'll just take you back to the MALDI-based imaging mass spectrometry strategies and how we use this as the discovery-based strategy to identify proteins that have what we call disease state distribution patterns, meaning that the abundance or the distribution of the protein is affected by the infected status of the animal. And so here again, we have a Staph aureus infected kidney on the left, and you'll see there's abscesses all throughout this kidney. And then on the bottom, we have a number of molecules that have disease state distribution patterns. I'm hoping you can see my cursor here, and you'll notice that some of these molecules have very interesting patterns. For instance, these thymosin proteins are occluded from the abscess. They're not present within the abscess, which is the exact opposite of S100A8, which is diffused through all of the abscesses. But this is very different than what we see, for instance, in this protein here, which is diffused through one abscess, but only rings the outside of the other. Or this protein here that is on the outside of all the abscesses, similar to that metal distribution pattern I showed you earlier, but not localized within them. And if you look carefully, you'll notice there is an abscess right here with no signal in it at all. So the point of these images, or the point I want to make with these images, is that it's very clear that the abscess has a molecular heterogeneity to it. And even two abscesses in the same tissue do not have the same molecular inventory. And so you can imagine if you're a bacterium and you're in, say, this abscess, you're going to have a very different environment that you're encountering than if you're in this abscess. And so when we just grind up this tissue and do proteomics or RNA-seq to look at bacterial gene or protein expression, we're losing a lot of information. We're losing that molecular heterogeneity. And we'll go through the talk and show you more examples of this, some of which that are related to metal exposure. In addition to being a discovery tool to identify new patterns within a single tissue, it's a very powerful tool to study systemic changes in proteins following infection. So Staph aureus can infect every tissue in a vertebrate, and if we infect an animal intravenously, every tissue within that animal will become infected, but we will only see lesions within the kidney in this model, as I mentioned. So imaging mass spectrometry can be applied to a whole animal to identify molecular changes that occur throughout the entire animal, even in the absence of overt pathology. So here we have a mouse that's been infected intravenously in section from nose to tail. And then on the bottom, we're imaging molecules that have a disease state distribution pattern, meaning in an uninfected animal, these would be black rectangles. So this protein here accumulates in the liver of the animal. We have proteins that accumulate in the kidney of the animal. This is a protein that changes abundance in the gastrointestinal tract following systemic intravenous Staph aureus infection, which is very interesting. This protein was also pretty striking. This is an eyeball protein, crystalline, and we've infected these animals retroorbitally. So we've either damaged the tissue or we've infected the eye as part of that process, but clearly we see a change in abundance of that protein. We have intra-organ specific differences. So here's a protein in the lumen of the stomach versus surface of the stomach. And then here, of course, is our favorite protein to image, which is calprotectin. So calprotectin accumulates in the kidney abscesses, as we've seen before. But you'll notice the second hottest region of the tissue is over here. And this was a very fortunate section for us because we sectioned through the femur of the animal. So we're actually imaging bone marrow here. And I think what we're imaging is calprotectin as a neutrophil protein in neutrophils that are developing in the bone marrow and are then going to traffic to the site of infection to combat Staph aureus. And this sort of gives you a sense for the systemic power of this imaging modality across an entire animal. Quickly, I'll introduce you to MRI, which is another technology that we use to image infected animals. This is just like the MRI that you or I would get. The only difference is the bore or the hole where we put the animal is of course much smaller since this is a mouse and not a person. And the benefit of MRI is it allows us to image a single organ over time without sacrificing the animal, and we can monitor tissue pathology and abscess formation. And we see by day 3, abscesses begin to form, and they're fully formed by day 4. And the reason I show you MRI is because one of the things my lab is most interested in recently is trying to perform multimodal imaging, where we merge different imaging strategies together to benefit from the strength of each modality. And so the question we wanted to ask at this point was, can we benefit from the anatomical resolution provided by MRI and the sensitivity and specificity provided by the mass spectrometer to apply both these technologies to a single infected animal. And so this is our first attempt at that. And the way that this experiment went was we infected mice intravenously with Staph aureus. We then imaged the animal with an MRI and the kidneys. We imaged the kidneys in 3 dimensions. So as we move back and forth through these either block face or MRI images, those white spots are the abscesses. We then took the animal out of the MRI and we deep froze it. And then we sectioned it every 100 microns. And every 100 microns, we took a mass spec image. And we made a 3D volume rendering of all the mass spec data and co-registered it with the MRI data. And the end result of that experiment is an image of the 3-dimensional distribution of calprotectin within an infected animal, here focusing on the kidneys. And we can, of course, do this with multiple proteins. Calprotectin is nice here because we think what we're actually imaging is neutrophil distribution in 3 dimensions throughout this kidney as imaged with a mass spectrometer looking at a single neutrophil protein. And we were excited that this experiment works so well because it really set the stage for the experiment we really wanted to do, was to combine even more imaging modalities to ask the question, can we use either MRI or CT to see how infection damages tissue? Can we use MALDI imaging mass spectrometry to image proteins in those tissues? Laser ablation inductively coupled plasma mass spectrometry to image elements in those tissues, and then do this when mice that have been infected with strains of bacteria that are driving luciferase off of metal-dependent promoters imaged using optical techniques. So the idea is, in a single experiment, can we ask, how does infection damage tissue? How does that lead to the recruitment or redistribution of metalloproteins? How does that affect metal abundance? And then how do bacteria change gene expression in response to that altered metal abundance? And here is an example of that experiment. It's a little hard to orient you without you seeing me in real life, but this is a section that's been taken through a transverse section of the animal. So the mouse has been cut in half. The easiest way to think about this is imagine that the animal were lying on its stomach where its head was facing you, tail was facing it, and we section the animal in half. We're looking at the entire animal. It's been infected with Staph, and you'll see a single abscess here. And in these first images, what we've done is we've co-registered the MRI data with elemental images for manganese and protein images for calprotectin in green and orange respectively. And I should just stop real quickly and say you'll only notice mass data accumulate in this region of the tissue, and that's simply because that's the only region of the tissue where we focus the mass spectrometer. So of course these molecules will also exist in other portions of the tissue, we just didn't use the mass spectrometer to image them. So as we move through, we'll see abscesses appear in the kidney. Those abscesses will be filled with calprotectin, as we've shown before. And manganese will also be in the sort of ring structure or the cortex of the kidney around the abscesses. Here we're looking at actually calcium. I got to go back, I'm sorry. So this is the manganese image. So again, calprotectin is in the abscess, and the manganese will be in this ring-like structure around the outside of the tissue. It's very similar to what we saw in 2 dimensions. We're just now seeing it in 3 dimensions. And the same is true for the calcium images. So here, calcium is localized within the abscess now. So calprotectin is orange and the calcium is blue. So you'll get calcium and calprotectin co-registering with this green color within the tissue abscesses. Again, these first 2 images are not that different than what we learned with a 2-dimensional image. I just wanted to show you, to orient you to what we're able to do with this technology. I mentioned that these mice were infected with a strain of Staph aureus that's driving luciferase off of a metal-dependent promoter. In this case, we're using the FIR promoter, which is a promoter that drives gene expression when Staph aureus is starved for iron. So this particular strain of bacteria will glow when it's starved for iron. And that glowing can be imaged using optical imaging strategies as these golden spheres within the tissue. And in the case of an optical imaging strategy, we're imaging the entire animal. So as we move through this animal, we'll see these gold spheres appear, and this indicates Staph aureus is colonizing the tissue and producing luciferase because it's iron starved. And a really interesting thing from this experiment is that everywhere we see an abscess, we will see a golden sphere, because that's where the staphylococci are. They're within the tissue abscess. But everywhere everywhere we see a golden sphere, we see an abscess. But everywhere we see an abscess, we do not see a golden sphere. In other words, everywhere that staph is producing light, it's localized within a tissue abscess, but not all staph within an abscess are producing light. And that means that Staph aureus is either differentially metabolically active within one abscess as compared to another, or alternatively, it's experiencing different levels of metal starvation in one abscess as compared to another. And the reason we think that is interesting and consistent with our previous mass spec data that I showed you with the MALDI images, where we can see that the abscesses have a very different molecular inventory. Again, all sort of telling us the story that Staph aureus within one abscess is not experiencing the same molecular environment as Staph aureus within another abscess. So now that we can image iron, we can ask, or image the bacterial response to iron starvation, we can ask, how does that relate to the localization of iron using the LAICP-MS? So in this experiment, the yellow color is iron as imaged using the ICP, and the red sphere is the luciferase signal. And everywhere we see large amounts of iron in the tissue, we do not see the bacteria producing iron-dependent gene expression. And where we see iron-dependent gene expression, it's in largely regions of the tissue that are not particularly rich in iron. Again, consistent with this notion that Staph aureus is experiencing molecular heterogeneity depending on where within the tissue it is localized. So those movies were fun to make, but this is actually our favorite image from this experiment, and it's not going to move. It may not seem that impressive. It's just this little dot here. But the reason that we were so excited about this is the first time that we were able to use a mass spectrometer to identify a bacterial protein in tissue. So this is the Staph aureus delta toxin. It's a hallmark Staph aureus virulence factor. You may wonder why it's so small in an otherwise large abscess. And the reason is because the amount of bacteria that generates an abscess is actually very small. So this is the microcolony responsible for the formation of this large abscess. So you'll notice that that mass signal for delta toxin perfectly overlaps the staphylococcal microcolony. One thing that I think is powerful about this image is it now tells us that we have the ability to identify bacterial proteins that are expressed in tissue, which I think has important ramifications for vaccine development and antimicrobial targeting. You want to make sure the antigen you're going after is actually expressed and abundant. And then the second is from a diagnostic standpoint, this is a hallmark characteristic Staph aureus protein. No other bacteria make this toxin. So the ability to image infected tissue and immediately identify the bacterial species using this strategy, we think has some potential applications for clinical microbiology. And knowing that we can image bacterial virulence factors, we can then go back to our calprotectin images and ask, how does a bacterial virulence factor's localization pattern relate to a protein of the innate immune system? And here calprotectin is the neutrophil protein imaged in blue, And the purple dot in the middle of that blue cloud is delta toxin. And an interesting thing that's sort of hard to show over Zoom through these movies is that the calprotectin protein, the blue signal, does not get all the way to the purple signal. There appears to be this region in between the delta toxin, which represents the bacterial microcolony, and the calprotectin, which represents the neutrophil, suggesting that maybe the neutrophil antimicrobial peptides or proteins are not getting all the way to the microcolony. And we're very interested in trying to identify molecular features that may be responsible for degrading or repelling neutrophil proteins that are trying to get to the microcolony to destroy the bacteria. I'll just put this last image in for those of you that aren't interested in innate immunology or infection, just to show you that we can image a number of different elements through healthy or diseased tissue. And this is just an example of a number of different elements, all color-coded in different ways, so that you can see not only does this technology provide the capability to image these elements, but it also shows pretty clearly that they don't all have the exact same distribution pattern as you would expect, but it's nice to see that in 3 dimensions. So those are some of our more recently published work, and now I would like to spend some time telling you where we're trying to take this technology. So this next part of the talk is focused on future applications of imaging mass spectrometry. Lillian was a student in the lab, as was Will Perry on the right. He was a chemistry PhD student. Anil was a postdoc, and then Raf is a really talented computational scientist from the Netherlands who runs his own independent research group. And Jeff is an extraordinarily talented mass spectrometrist who has his own research group here at Vanderbilt. And so I've shown you images to date that we've imaged infected tissue at approximately 50 microns. And 50 microns can generate a lot of valuable information for us. So we see pathogen-specific proteins now, we see tissue-specific proteins, innate immune proteins. But the really exciting thing is that recently Jeff and Raf and their team have gotten a lot more powerful or a lot higher resolution in their imaging. And so here's an example of an image they have taken of a rat brain at 10 micron resolution. And we're imaging 5 different masses on the left. And hopefully you'll appreciate that the resolution of the mass spectrometer now approaches that of the microscopic stain on the right. But the difference is on the left, you're actually getting molecular information, whereas on the right, it's a fairly non-specific stain. So this high resolution imaging is something that we've recently begun applying to infected tissue. And I just wanted to show you some of our best images and some of the ones that are most relevant to this group. First, this is one of our best images. This is, again, Staph aureus infected kidney. The abscess now is this large thing shown in pink. And here is otherwise healthy tissue surrounding it. And you'll notice this yellow signal here is a bacterial specific signal. This is a lipid modification called lysyl PG. And that's a modification that Staph aureus has been shown to produce in vitro as a way to change the charge of the bacteria to repel antimicrobial peptides. Interestingly, that modification had never been observed in vivo, just of course predicted to occur. And here, using a mass spectrometer, we can identify this modification occurring within select bacteria within the microcolony. There's other bacteria within this abscess that are not making this modification, again, further supporting molecular heterogeneity across the staphylococcal microcolonies throughout even a single tissue abscess. So to date, or at this point in the talk, every molecule, bacterial molecule that I've shown you localizes exactly on top of the bacterial microcolony. So we're really interested by this mass here, which is also a bacterial molecule, but you'll notice it does not localize to the microcolony, and instead it's very diffuse throughout an entire abscess. And we identified this molecule to be the bacterial siderophore staphyloferin A. So these, of course, are small molecules that are secreted from bacteria where they bind iron with a very high affinity to provide the bacteria with a nutrient iron source. And it makes sense to us that these molecules would have a distribution pattern that was much more diffuse because this is a molecule that exists to scavenge nutrients and provide them back to the bacteria. These molecules are produced when Staph aureus is iron starved, and you'll notice that not every abscess has large amounts of the staphyloferin A being produced. Again, supporting this notion that staph is not uniformly iron starved across abscesses and presumably not experiencing uniform molecular environments across abscesses. Just a few more images of siderophore imaging. These are infected kidneys again. We're imaging staphyloferin A or staphyloferin B. Clearly, you can tell that not every abscess is full of these siderophores, and we can image them in relation to iron as well on the right, again, integrating our multiple imaging mass spectrometry strategies. This is an infected heart. We have a kidney abscess here. That abscess accumulates these staphyloferin A and staphyloferin B siderophores. In this case, we have a strain of Staph aureus that produces GFP when it's iron starved, so you can see that it's producing, where it's producing the siderophore, it is iron starved, and there's no detectable iron in that abscess. And this is a nice image, I think, that shows how we can, in 2 dimensions, use all these imaging modalities to really image the struggle for metal between host and pathogen. And then this is, again, in liver, a very similar image. So just a preview on the next 2 slides. We've just finished, or recently have finished, the collection of a 3-dimensional data set through a kidney of an infected animal at 10 microns. And we're hoping to submit this paper within the next few weeks. And I just wanted to show you a few movies that we put together quickly so that you can see what the images look like. So on the left, we have autofluorescent microscopy, and then on the right is the imaging mass spectrometry data at 10 microns in 3 dimensions. Here are 3 separate masses to give you a sense for the resolution that we're getting at 10 microns throughout the entire organ. And then finally, this is just a tissue abscess here. And this one, again, I think I have to click them. This abscess on the left is also at 10 microns, and we have a tremendous amount of molecular information now that we're sorting through, trying to identify molecules that have really interesting disease state distribution patterns in multiple dimensions now. And again, we're submitting this paper soon, and so hopefully, as we synthesize the data, we'll be able to tell some interesting stories about some of the molecules that we've identified. In just the last few minutes, very quickly, I want to talk to you about where my lab is heading with some of these imaging technologies, and that is to look at microbe interactions in the context of metal restriction. And the reason that we became interested in this is this appreciation that bacteria are thought to largely exist in the environment, regardless of the environment that we're talking about, in the form of communities, or microbial biofilms is a more commonly used term. And biofilms it's hard to study the heterogeneity within a biofilm for a few reasons. If you want to do, say, proteomics or RNA-seq, you have to destroy the biofilm's architecture, and so you lose information on spatial heterogeneity. If you want to use labeled molecules or antibodies, those can be very powerful, but they again require some information about the molecules you want to image. You have to have the reagents in hand, or you have to specifically know the molecules that you're trying to target. So we wondered if we could use imaging mass spectrometry in an unbiased way to image microbial communities and study how metal restriction affects those communities. And this work was done by Kathy and Mike. We're postdocs in the lab, and I've already met Yao Feng and Jessica multiple times. And so for this, we use Pseudomonas aeruginosa. I'm not going to go into any background on this organism. Professor Nolan may introduce us to some Pseudomonas because she's done some beautiful work on that organism. I will instead just say that we use this organism just because it makes really good biofilms. And we wanted to show proof of concept that we could apply this technique to microbial communities, so we picked an organism that made a really thick biofilm. We use what's called a biocell, or a flow reactor, where a microscope slide is placed in the reactor and media is dripped over the microscope slide over the period of days until this large community forms that a microbiologist would say is beautiful. A non-microbiologist in the audience might not agree with that, but then you can pick this thing up with tweezers and we can section it just like you would section tissue. And so here's a section of the biofilm before we put it in the mass spectrometer. So to orient you to sort of the anatomy of the biofilm, the liquid drips in here. We call it the nutrient-rich pore, and then the region in the middle is the most nutrient-rich region of the biofilm. And as the biofilm gets further away from the nutrient channel, you get more nutrient deplete edges. And so we would then take this biofilm and we section it like you would section a loaf of bread. And what I'm about to show you is if you imagine a piece of bread and I'm now turning it to you so that you can see the molecular features. And so here are some high resolution imaging mass spec images of a bacterial biofilm, Pseudomonas biofilm. we just focus on section 1. Again, we're looking at sort of the piece of bread held towards us. And this is the nutrient-rich portion of the biofilm where the liquid flows, where the media flows. And you see there's some very distinct molecular features that are different than molecular features that we detect at the nutrient-poor edges of the biofilm. And if we put a bunch of these masses together in a single image, I think what becomes immediately obvious is how much remarkable molecular heterogeneity exists even within this single microbial community of a single microbial species. And so what we've begun to do is to ask how elements are distributed throughout this community with the hope of learning how elemental fluxes or changes in elemental gradients impact microbial protein production within a community. And so here's one of our first images, the LA-ICP-MS images on the left. We don't yet have the ICP-MS images at the same resolution of our sort of MALDI or FTICR based images. And so that's why they're a little grainier, but I think you can appreciate that the nutrient-rich portion of this microbial community is where we see the most manganese and zinc. And we don't see that at the edges, but iron seems to be more diffused through the whole microbial community, suggesting there may be molecules that are more easily diffusible, that bind to iron, that can move iron throughout the microbial community, which is different than what we see for manganese and zinc. And this bottom image is just to orient you to the nutrient-rich central channel as compared to the nutrient-deplete edge. So in the future, we plan to use this technology and integrate it with some of the other optical imaging technologies that I introduced you to earlier to study how microbial communities change when they're exposed to metal stress, be it excess or deprivation. And we'll do that by modifying what we put through the flow cell system. So just to summarize at this point, my lab is interested in this intersection between infection and nutrition. We primarily focus on nutrient metals, and we do a lot of work with the staphylococcal abscess because it's this walled off battleground where the immune system and the bacteria are fighting for these nutrients. And we think that that hostile environment has led to the evolution of a lot of really sophisticated strategies and also allows for some really powerful imaging technologies. And so we study this with some of these more conventional molecular microbiology methods like I introduced you to on the right, as well as others. And then we'll also use some more advanced mass spectrometry-based imaging technologies. And in the case of the calprotectin story that I told you, which again, we're about to hear a lot more about, I think, from Professor Nolan, we know that neutrophils pour into this abscess to fight Staph aureus, but a hallmark of Staph aureus is the ability to make leukocytins. These are toxins that can lyse neutrophils. And we think what calprotectin is a posthumous antimicrobial strategy within the neutrophil that once the neutrophil is lysed by Staph aureus, the abscess fills up with this antimicrobial protein that binds up all the available metal and restricts microbial growth. And we continue to study that protein and its contribution to infection in my lab. And so that, I'll just acknowledge we are fortunate to have a large group of collaborators. This imaging project in particular in my lab is a very interdisciplinary project. I tried to acknowledge the major contributors as we went through the talk, but some more of them are listed here. And then also I tried to acknowledge my lab as we went through the talk. The current members are on the bottom and the past are on the top. And so I won't spend any time individually acknowledging them, just say that I'm very fortunate to have such a great research team. And again, hope I acknowledge everybody as we went throughout the talk. And I just want to thank Sega and Eva again for putting this meeting together, particularly under this complicated situation. So thank you for your attention. And if you have any questions, I would be happy to answer them.
Okay, thank you, Eric, for a wonderful talk. I think it was extremely interesting. So I'm sure we will have some questions, but I think maybe I can start with